Hello everyone, this is Professor Youssef, and today we are going to be discussing the urinary system. We'll be talking about many of the very important anatomical structures that contribute to the urinary system, such as the kidneys mainly, and some of the more accessory organs that we see downstream. We'll also be talking about the histological identifications that you should be familiar with for your practical exam. And then we'll briefly look through the MINC and some different structures that you might want to be looking out for as you study. And just please bear with me with the audio. I know it might sound a little bit tinny in some areas. The recording environment I'm in now is not my usual one. But the content is all there. So, I'll do the best I can in the post-editing stage to get that, as tolerable as possible. But for now, let's just jump right into it and start talking about the urinary system. So as a lot of us are already pretty familiar with, the urinary system is largely going to be centered around two very important paired organs called the kidneys. These are going to be located on either side of the vertebral column. And they're considered what's called retroperitoneal. Now, we mentioned the hypochondriac space and the retroperitoneal area in AMP1 when we talked about the different anatomical orientations and different regions of the body. And retroperitoneal simply means that it is behind the peritoneum. We know that the abdominal cavity is going to be lined with this serous membrane that we call the peritoneum, right? It's going to hold those abdominal organs. And there's going to be a visceral, parietal peritoneum, just like any of the sacs that we talked about, right? The pericardium, for example. So the peritoneal cavity, as we know, over here. And the kidneys, we're going to find them just behind that peritoneal cavity right over here. Okay. So as we know, the kidneys are going to be very important for several different reasons. We know that metabolism of proteins, for example, is going to, proteins, of carbohydrates, of all different types of macronutrients is going to produce waste, right? And in doing so, we're going to find waste products like carbon dioxide, nitrogenous wastes, right? Ammonia, urea. And these things need to be eliminated if the body is going to function properly. So these excretory processes are going to involve multiple different organ systems, and the primary one is going to be the urinary system, at least with regard to the removal of nitrogenous waste from the body. In addition to this, the excretory function of the kidneys is not all that it does. It also has importance when it comes to maintaining electrolyte and acid-base balances, fluid balances of the body. So it's really a major homeostatic organ. It has a lot to do with the homeostatic processes that take place to just make sure everything's in equilibrium, everything's in balance. And in order to do that, the kidneys have to first act as what we would probably think of as just a blood filter, right? So it's kind of like a blood filter, and then it's going to act as a filtrate processor. Now, the filtrate is the stuff that gets filtered, right? And essentially, it's going to filter everything and then pick and choose very specifically what it wants to bring back into the body, right? So think about when you're cleaning out your refrigerator, right? You don't only take out the stuff that's spoiled, you take out everything, right? You take out everything and you put the stuff back in that is still good, and you throw out all the other garbage that went bad or is rotten or whatever, right? So that's kind of how the kidney's going to work. So it's going to allow toxins, metabolic wastes, too many excessive ions, right, to leave the body in the urine, and then is going to pull back any of the substances it really needs by returning them to the blood. And that's going to happen via the nephron. The nephron is the main functional unit of the kidney. We're going to look more closely into that in a little bit. And it's clear to see pretty much why malfunction of the urinary system, particularly of the kidneys, like we might see in chronic kidney disease, for example, that malfunction can really lead to a major failure in homeostasis, which, unless corrected, can be undoubtedly fatal, right? So you definitely don't want a buildup of those toxins, and you definitely don't want to not be reabsorbing those important substrates that your body needs. Instead, you don't want to be peeing those all out at the end. So pretty important functions for the kidney. The remaining organs of the system, like the bladder, the ureters, urethra, these are all going to function to store urine, right? They're going to be temporary storage reservoirs or transportation channels for urine, and we'll get into those as well. So there are a few important key structures you guys should be aware of when it comes to the kidney. We'll start with the kidney, of course. And the internal anatomy is relatively straightforward, but there are a few nuances you should be aware of. So the first thing I want to bring to your attention is the fact that there is a cortex and a medulla. Like we've seen in many other systems before, there's a cortex and a medulla in the kidney as well. Now, cortex referring to the cortical region, the outside region over here. We can see the cortex. That's going to be the outer portion. We have a capsule type of tissue, connective tissue surrounding all of that in the outside, the renal capsule. And inside of the cortex, we're going to see the renal medulla, Now, you can see this both in the organ and histologically that the cortex, the outer tissue of the kidney, is going to be a little bit lighter in color. It's not going to be as dark red as we see in the medulla, right? And if we look closely, we can see that this cortex, while mostly on the outside, it's going to kind of dive into this renal medulla area, right? Causing these little different subsections. And these are going to be called medullary pyramids. And these little areas where they dive in are going to be referred to as renal columns. So here's our renal columns. Okay. And if we kind of zoom in a little bit on this one little square portion that we see over here on the left, we're going to see a little bit closer, zoomed in, that there's going to be what are called nephrons. These are these tube-like structures that are going up and down throughout the medulla and the cortex. And we'll soon see that there are certain structures that are only present in the renal cortex and others that are going to be in both, right? Like the collecting ducts and the different descending and ascending loops of Henle. Those will be in both, but the glomeruli, for example, which we'll talk about, are going to be only found in the renal cortex. And we can get a little bit more of a close-up view over here all the way on the right of the individual functional unit called the nephron. Now, before we dive into some of the more nuanced structures of the kidney, I do want to bring your attention to a structure called the renal hilum. And this is going to be the entry and exit site for structures servicing the kidneys, right? So we can see those over here. That's the renal hilum is going to be right in this area. And it's going to be letting things in like vessels, nerves, lymphatics, ureters, and that's obviously pretty important when it comes to the whole entire filtration process. So the medial facing hila, the plural for hilum, hila, are tucked into this sweeping convex outline of the cortex, right over here, the inside part. So emerging from the hilum, we have what's called the renal pelvis. The renal pelvis is all this area here, and that's going to be formed from what are called the major and minor calyces in the kidney, right? So calyces is plural for the word calyx, or calyx, however you want to pronounce it, right? So if we look over here, we could see the minor calyx. These are the smaller structures that are kind of feeding out from the renal pyramids. These are the renal pyramids that we saw, right? We talked about the renal columns and the renal pyramids that are formed as a result. So that's a minor calyx. And then the major calyx is going to be quite a few of these minor calyces pretty much merging into a major calyx. Okay. And there's also going to be some smooth muscle in the renal pelvis, and that's going to be funneling the urine via peristalsis, just like we saw in the digestive system, into the ureter. Now, we don't really see much of that smooth muscle in here, but it is in there. Just so you're aware. And then also we have the renal arteries and the renal veins, right? We can see the renal arteries are forming directly from the descending aorta. If we look over here in the top left, we can see that this descending aorta. and we can see the vena cava as well. So we have the renal arteries. They're going to be coming from the descending aorta, whereas the renal veins are going to return that cleansed blood directly to the inferior vena cava, which we know is going to empty back into the right atrium of the heart. Okay. So these artery, vein, and renal pelvises are arranged in an anterior to posterior order. Okay, so you can see the artery, vein, and then you see the renal pelvis in the back, the posterior aspect. So briefly, let's just cover what the general flow of urine looks like. All right. So after we move through that functional unit, all those thin tubes that I showed you earlier of the nephron, urine is ultimately going to drain from these renal pyramids. They're going to drain into what's called the renal papilla, right? Here's the renal papilla over here. Here's a bunch of them. From the renal papilla, we're going to see them go into the minor calyx, right? Here are some minor calyces, right? Every renal papilla leads to a minor calyx, and we know that many of these minor calyces are going to form what's called a major calyx, right? Those big ones over here. And ultimately, all those major calyces are going to drain into this whole renal pelvis that we talked about earlier. And then from there, out of the renal pelvis, we're going to see the urine kind of drop into this ureter, this tube-like structure leaving the kidneys, which will eventually enter the bladder, and then from the bladder will leave the body via the urethra. So as you guys know, this is an anatomy course, and your practical is going to be strictly on the anatomical function, so I'm not going to get too much into the physiological aspect of things. Dr. LaCroix-Freilich will definitely show you more of the nuanced mechanistic processes that underlie these structures. But for now, your job is to be able to tell me what something is if I point to it, right? So let's just go ahead and talk a little bit about the vasculature of the kidney, right? So we know that the renal artery is not going to stop there. The renal artery is going to divide into smaller arteries and then further branch into even smaller arteries, and then they're going to, eventually become arterioles, which will then transition into the more vein-like structures, right? That are going to carry the filtered blood back. So let's just run through those really quickly. The renal artery here, which we see entering this hilum, right? That renal artery is going to first divide into what are called segmental arteries. Segmental arteries are going to be kind of in this area over here within the pelvis, okay? And that's going to be followed by further branching to form what are called interlobar arteries. Interlobar arteries. We're going to see those up here in the lobes, right? And they're going to pass through the renal columns to reach the cortex. And these interlobar arteries are in turn going to branch into what are called arcuate arteries. And these arcuate arteries we're going to see turning into what are called cortical radiate arteries, aka interlobular arteries, right? So they're going to be within lobules. And then finally, they're going to differentiate into what are called afferent arterioles, right? Remember the difference between afferent and efferent? These are going to be afferent arterioles, and they're going to service about 1.3 million nephrons in each kidney. Okay, now if we look a little bit closer, here we have the afferent arteriole on the right, and that is going to be leading into this bundle of structure of capillaries called the glomerulus. Glomerulus comes from the Greek bundle of yarn, or a ball of yarn, right? And it kind of does look like that when you look at it from a histological perspective. So after we are done with this whole bundle of yarn complex, this glomerular structure, and we leave the glomerulus, then we have what are called efferent arterioles. Efferent. So afferent at, efferent exiting, and so the main point of reference that we're referring to is going to be the glomerulus, right? So when we were talking about neuro, for example, right, the brain, we knew that afferent neurons were going to be toward the brain and efferent were leaving the brain, or the spinal cord, the CNS, right? So in this case, the CNS, the brain or spinal cord, is going to be analogous to the glomerulus. Okay, so that's our main point of reference. So after we we leave the glomerulus as our efferent arterioles, they're going to break up further into what are called peritubular capillaries. And these peritubular capillaries are going to be encompassing or surrounding the entirety of the nephron. This whole thing that we're looking at here is the nephron, right? It's going to go through all different parts of the nephron, and this is what allows for the transport of solutes to and from, or rather from the specific tubes, right? There are different tubes here, the proximal convoluted, distal convoluted tubule, the descending and ascending loops of Henle, right? We have all these different tubes, and they're going to all be allowing certain things through, right? Every section of the tubule of the nephron is going to be permeable to very specific substances. So these peritubular capillaries are there just so that there's easy transport from the nephron into the blood circulation, okay? Now, at some point, we're going to see a transition into these venous structures, right? We're going to be leaving the peritubular capillaries and entering into what are called our interlobular veins, right? So we have an interlobular vein, and that interlobular vein will become an arcuate vein, then an interlobar vein, then a renal vein, right? There's no segmental vein like there is in the a segmental artery. So let's just run through these really quickly. Arterial blood flow in the kidney. So as we know, abdominal aorta will branch into the renal arteries, right? The renal arteries are going to give way to the segmental arteries, which we're going to see over here, coming right off. And the segmental arteries are going to then break up into our interlobar arteries. We can see those over here, interlobar arteries. Okay. They're going to be going up through these renal columns, and they're going to become arcuate arteries. Arcuate arteries we're going to see kind of projecting up here into our cortex. Those arcuate arteries will become what are called interlobular arteries, which are going to be proceeding directly up into the cortex, right? Even further, those arcuate arteries will then become, or those interlobular arteries will then become afferent arterioles, right? So again, we have to get a little bit more microscopic. If we look up in here, those afferent arterioles are going to be going and entering into our glomerular capillaries, right, in that glomerulus. They'll be leaving as efferent arterioles and then eventually becoming peritubular capillaries, which are going to be surrounding that nephron. From there, we are going to have our venous blood flow, right? So from our peritubular capillaries, which we see surrounding the nephron, we're going to see those differentiate into what are our interlobular veins, right? So we're kind of just backtracking now. Our interlobular veins will then become our arcuate veins, which we can see over here. Those arcuate veins are going to become our interlobar veins, and those interlobar veins are going to become the renal vein that we see entering into the hilum. Okay, notice we don't have a segmental vein. We only have that on the arterial side, a segmental artery. There is no segmental vein, so, a little bit less to know. And then, of course, right from the renal vein, we're going to go into the inferior vena cava, which will then ultimately dump into the right atrium of the heart. Okay, guys, so let's take a closer look at this glomerulus that we were talking about earlier. The glomerulus, as we know, is going to be this bundle of capillaries, okay? And we're going to see these afferent and efferent arterioles entering and exiting, respectively, from the glomerulus, right? And there's a few important structures you should be aware of when looking at the glomerulus. So first of all, I do want to point out this glomerular capsule, right? Also known as the Bowman's capsule. You will hear the Bowman's capsule quite frequently. It's a pretty common term. And that Bowman's capsule is going to be kind of surrounding the glomerulus and leading into the beginning point of the nephron, which we know as the proximal tubule. The proximal tubule, remember, we know what proximal and distal means by now. Proximal meaning the closest to, distal meaning furthest from. So the proximal tubule will be the tubule that is closest to that reference point, that glomerulus, okay? So now that we have that down, just be aware that the glomerulus is going to be strictly referring to the bundle of capillaries. The glomerular capsule, aka the Bowman's capsule, is going to be surrounding the glomerulus. There are two different things. Okay. Okay. And just so we're all aware, the renal corpuscle is going to be both of those together. Okay, so the glomerulus and the Bowman's capsule put together, right? So all of this business is going to be our renal corpuscle. Do not get these confused, okay? Because, you know, if I'm only pointing at the inside portion of this whole renal corpuscle, I'm talking about the glomerulus. If I'm talking about that ball of yarn, those capillaries, okay? If I'm pointing only at the outside portion here, right, this capsule around, I'm looking for Bowman's capsule, okay? If I say, what is this whole structure, then renal corpuscle makes a lot more sense. Just so we're all aware, because those three terms, unfortunately, they get mixed up quite a bit. Okay, now let's talk about the afferent and efferent arterioles. Okay, so we know that afferent, with regard to the Bowman's capsule, with regard to the glomerulus, is going to be leading toward the glomerulus, right? We're going from all those arteries and arterioles that we talked about, we're going from there into the glomerulus, hence afferent at the arteriole, at the glomerulus, excuse me, right? And we'll see eventually that these are going to be touching the distal convoluted tubules. We don't have a good view right now of the whole nephron, but we'll take a look at that in just a moment. Okay. Actually, let's go right over here to this next slide. I'll show you really quickly what I mean. So here's that afferent arteriole, right? We can see the glomerulus, all these. Here's the proximal tubule we talked about. It's going to go down into this loop of Henle, come all the way back around, and now this is the distal tubule. Right? So technically, in terms of the tube, it's further away. And we're going to see that that afferent arteriole does have contact with the distal tubule. Okay. That'll make a little bit more sense as we get closer. So the efferent arteriole, we're going to see is going to be exiting the glomerulus. Now, it's important to be able to differentiate between the afferent and the efferent, and the easiest way for you guys to do that is simply the diameter of the vessel. You'll notice that in just about every case, the afferent arteriole is going to be much larger in diameter, right? It'll be a bit thicker, it'll have a larger lumen, a bit more blood flow going through it, right? And the efferent will be a little bit smaller in diameter. And there's a reason for this, guys. You know, so I said I wouldn't get too much into the physiological side of things, but it's interesting and it kind of ties into what we're talking about here. By having the afferent arteriole larger, right, in diameter, that means we have more blood flow going into the glomerulus. By having the efferent arteriole smaller, we have less blood flow leaving. What does that mean? That means we're going to have a lot of increased pressure here within the glomerulus. Within that Bowman's capsule will be a lot more pressure. And that's what we want to see. That's kind of the driving force that is pushing all of this stuff out into our nephron. That's how we leave the blood vessels, leave these capillaries, and enter into the nephron. Right? Because it's pushing, you have all that pressure pushing the stuff out of the vessels, and that's very important. That's how we get that transition, right? That's how we get that movement of plasma and different types of very small substrates like, gases and electrolytes and, and amino acids, for example, into our urinary filtrate, right? So just a little side note. Anyway, let's zoom in a little bit over here on this image on the right. Podocytes. Podocytes are something that you should be aware of. These are found within the Bowman's capsule, right? And they're going to be also surrounding the glomerular capillaries. Okay, now podocytes are cells, and they are going to be kind of surrounding all the, you can see over here, they're surrounding all of these glomerular capillaries, right? And they have these little long extensions called pedicels, right? They're like foot-like processes, kind of, and they interconnect with each other. They're like fingers that are kind of like, when you put your fingers together, like you're praying, for example, right? You put your hands together, and they kind of form that junction of all these pedicels connecting with one another. All right, so they're going to wrap around the glomerulus, and they're going to form kind of a filtering sleeve, right? So these are going to have little tiny holes in between these pedicels that are going to kind of function as a filter, right? We also have what are separately, these are different, by the way, from the podocytes and the pedicels. We have what are called fenestrations, and these fenestra, right, are holes within the glomerular capillary. So if we look closely over here, we can see a glomerular fenestra over here. Fenestrations. They're literally just holes on the inside and leading to the outside of the capillary itself. Literally, it's like you took a thumb pin or a thumbtack and just poked holes in your capillaries. Okay, and that is kind of allowing a path for these different substances to leave the capillary and enter into the filtrate, right? The urinary filtrate that's entering into the nephron. Okay, if we look a little closer into this electron micrograph, these are so small, by the way. These are so tiny you can't even see them at all using a regular microscope. You have to use an electron micrograph, which is that black and white image you see on top, to be able to even visualize them. And if we look up there, we can kind of see these podocytes with their pedicel foot-like processes extensions kind of popping out. Okay, so let's move on now into the real functional unit of the kidney, of this whole filtration process, right? This whole detoxifying process, excretion process that we're talking about with the urinary system. And that is the nephron, okay? And these nephrons, as you can see, are these windy, loopy, turny tubes, basically, right? That's basically all they are. They're just tubes, hence the term tubule, as you'll learn as we go on. But they are going to be these literal tubes that are specialized in certain areas of that entire tube to reabsorb certain things and not others, right? And there's a very, very complex mechanism underlying the reabsorption of certain solutes like salts and urea and water, and there are different hormones that affect the ability of these tubes and ducts to reabsorb certain substances. But we're not going to get too much into that again. But I do want to point out the main structures. You should be able to point out if I were to, for example, pull up a model, right? Because this is kind of hard for me to test you histologically on a microscope. Granted, you can, there are ways to do it. When I took my histology course at New York Med, they were definitely asking us, under a microscope to distinguish between the distal tubules, the proximal tubules, collecting ducts, and certain parts of the glomerulus, the juxtaglomerular apparatus. There's a whole lot of nuances that, fortunately, we're not going to torture you guys with. Just the main structures, right? So let's start with the two main types of nephrons. The two types of main types of nephrons. Nephra, right? We know that like a nephrologist, for example, is going to be a kidney doctor, right? Epinephrine, right? We talked about the catecholamine responsible for our sympathetic nervous response, or AKA adrenaline. Epinephrine on top of the right? The kidney, right? So renal, nephro, these are all going to be terms associated with kidney function. Okay, so these two types of nephrons are going to be our cortical nephrons and our juxtamedullary nephrons. Okay, and these are simply referring to where the how far they're going to span throughout the kidney. So the cortical nephrons, cortical, right? This is going to be in reference to the cortex. We talked about the renal cortex, that outer lighter colored layer of the kidney. So these are going to be nephrons that exist solely within the cortex, right? We're not going to see them extending into the renal medulla, okay? So we have the cortical nephron, like over here on the right, we could see, and you're going to have your glomerulus, you have your afferent and efferent arteries going in and out, and the entirety of it, including this lower bottom part that dips down called the loop of Henle, that's all going to be within the cortex. Now, we also have what are called the juxtamedullary nephrons, and these are going to have a much longer loop of Henle, right? We're going to see this loop that's going all the way down, the descending and ascending loop. Sometimes it's called the nephron loop, more commonly called the loop of Henle. And that's going to be dipping into the medulla, okay? Into the medulla, much deeper in. There is a reason for this, and for these, we have some in the cortical region, some are going to be juxtamedullary. We won't get too far into that, but there is a reason. You can feel free to ask me later, and I'd be happy to expand on it. But for now, let's just go through the main parts of a nephron. And these parts are going to exist regardless of whether it's a juxtamedullary nephron or a cortical nephron. They're pretty consistent all throughout. So we talked about the glomerulus, and we know that surrounding that glomerulus, that ball of capillaries, we're going to have what's called the glomerular capsule, aka, and more commonly known as, the Bowman's capsule. And that's going to be right over here, this big circular kind of pocket holding all of these capillaries. That's going to be our Bowman's capsule, okay? Then we have what's called the PCT, right? And that's short for the proximal convoluted tubule. Now, proximal meaning closer to the glomerulus, right? So that tubule is going to be, obviously closer in not necessarily proximity, because the distal is pretty close as well, as you can see, but it's going to be the first part of this, what is basically a long giant tube, right? So it's that first part. So we talked about the small intestine, for example, right? And the proximal duodenum, or the proximal ileum, for example. That's the part that's closest to the stomach. It's all one big tube, even though all of it's all kind of jumbled up, right? Who knows which, actually is proximally closer, but in terms of proximity. But I don't want to play with language too much, but you guys get the point. So we have our proximal convoluted tubule, convoluted just meaning kind of like, very twisty, turny, convoluted, right? Not very straight to the point, right? And that is eventually, notice the thickness, by the way, of these different tubules as we go through. That is eventually going to dip down and turn into what's called the loop of Henle. Okay, so the parts that are going down and up, and that little loop at the bottom is collectively known as the loop of Henle. So you're going to have what are known as descending limbs and ascending limbs. Descending, obviously, being our the parts of the tube going downward toward this loop, and the ascending limbs are going to be the parts going upward, away from the loop. Okay, those descending limbs are going to have different portions. So the descending limb, for example, is going to have a thick descending limb and a thin descending limb portion. So this is all the descending limb, but this is the thick descending limb, and this part is the thin descending limb. And the same thing is the case with our ascending limbs. We have a a shorter, thin ascending limb, right? So the thin descending is going to be longer than the thin ascending. We also have another thick ascending limb, right? So glomerular capsule, proximal convoluted tubule, thick descending limb, thin descending limb, loop of Henle, ascending, thin ascending limb, thick ascending limb, right? And that is eventually going to lead into what's called the distal convoluted tubule. Makes sense. We had a proximal, now we must have a distal. Here's our distal convoluted tubule. And then ultimately, if we look over here on the right, we can see that this distal convoluted tubule is going to ultimately drain out into what's called the collecting duct. The collecting duct is going to be these long, giant, ducts, right? They're going to be in big tubes of their own that are going to be leading all the way down to the renal pelvis, which will eventually empty out into the minor and major calyces, and then eventually the pelvis, and then eventually into the ureters, right? Okay. Now, I kind of just added this slide just kind of as an FYI for anyone who is interested in kidney function, or maybe just as a little preface to what you may learn or may have already learned in Dr. LaCroix Freilish's section. Juxtaglomerular apparatus. This is a very important kind of system that we have that is important for the regulation of our what's being absorbed, our blood, tubular fluid pressure, right? It's going to help with renin secretion, smooth muscle cell contraction. I really won't get into it. It's actually quite complicated once you get into it, but this is just a little kind of idea, just running through the general, purpose of the juxtaglomerular apparatus. If you are looking through it in different resources, or you're trying to study through different resources, you're going to see this come up a little bit, but it's going to be made up of two groups of cells, the macula densa cells. Those are going to be in your distal convoluted tubules. They're going to be monitoring your tubular fluid for low blood pressure. They're going to alert your juxtaglomerular cells, which are your second group of cells in the JGA, the juxtaglomerular apparatus. And these are just epithelial cells, right? And the juxtaglomerular cells within the afferent arteriole are going to be helping to secrete renin, right? Renin is ultimately going to help with the elevation of blood pressure, right? Because that increased blood pressure is going to therefore increase the amount of pressure in the afferent arteriole entering the glomerulus, and therefore will increase the amount of filtered product leaving the capillaries and entering into the Bowman's capsule space, that space, right? And it ultimately also has a lot of smooth muscle cells that are going to help with the contraction of the actual vessels themselves, right? So you can kind of contract the different arterioles and regulate the amount of pressure that you have within that glomerulus, right? And it's going to, be very finely tuned based on what the body needs. But I digress. Let's move forward to the next slide, which is ultimately leading away from the kidneys now into those different storage reservoirs and urine transportation systems we talked about. So we're going to start with the ureters, right? So we have the kidneys up here. We saw the kidneys. Here are renal pyramids. Here are minor and major calyces, our renal pelvis, and they're ultimately going to be leaving the kidney and entering into these long paired tubes that we call the ureters. Okay? Now, don't get these confused with urethra. These are different. The ureters are going to be paired. The kidneys are paired. The bladder, however, and the urethra are going to be single organs, right? They're going to be only one, right? And the urethra is leaving the bladder. Ureters are leading toward the bladder. Okay? So we have these two paired ureters leaving the kidneys, entering the bladder, and they're going to contain what's really interesting, and a pretty common feature of a lot of these structures that we're going to see moving forward. It's called transitional epithelium. And we talked about this before. We talked about transitional epithelium way before in AMP1. And we'll get a little bit more into it, but it kind of is a type of stratified squamous tissue that changes shape. Those top few cells are going to change shape based on the state of the organ that they are lining, okay? That'll make more sense as we move forward. But we're also going to see a nice couple or maybe even 3 layers of smooth muscle. Okay, so in the ureter, for example, specifically, if we move on to the next, actually, you know what, we're going to hold off on that until histology, but let's move on to the next slide into the urinary bladder. And if you look at the urinary bladder, we're going to see that it's an expandable muscular organ in the hypogastric region of the lower abdominal pelvic cavity, and it's going to receive urine from the ureters. You can see those 2 ureters up here, and temporarily store the urine until we're prepared to expel it, right, via excretion. So the wall of the urinary bladder is able to greatly stretch, right? Kind of like the stomach, right? It's capable of great distension. It can be stretched as it fills with urine. So the actual size and shape of the urinary bladder varies pretty greatly depending on the volume of urine it contains. So when the bladder is empty, we're going to look at the mucosal layer, right? And this is what I was talking about earlier with that transitional epithelium. When the bladder is empty, the mucosa will exhibit distinct folds known as rugae, right? We saw these rugae in the stomach as well. So, oh, interesting. Okay, so these rugae that we're looking at over here are going to kind of stretch as the bladder fills up and flatten, which allows the lumen of the bladder to expand as it fills with urine. We'll take a look at that a little bit more closely as we get into the histological side of things. But if we look over here, for example, on the bottom right-hand corner, we can see that in an empty bladder, we have these little dome-shaped epithelia on the top, right? They look a little bit more like domes, right? And that's in an empty bladder. But when we fill the bladder with urine, right, and we'll say the lumen is down here, and that lumen's getting bigger and bigger and bigger as it stretches and fills with a higher volume of urine, we're going to see that these dome-shaped top epithelial cells are going to actually kind of flatten and look a little bit more squamous in nature, right? So they're going to be a little bit more flattened, more scale-like, and that's going to kind of be an indication of whether we have a full or an empty bladder, okay? Again, we have smooth muscle in the urinary bladder, and that is largely contributing to what's called the detrusor muscle. The detrusor muscle is going to be what contracts the bladder. That's going to be allowing for the expulsion of urine from the bladder into the urethra and ultimately out into the environment. So here's that detrusor muscle. You can see it's a very thick muscle, and that'll be one of the distinguishing factors histologically that allow us to distinguish it from the ureters, for example. And you're going to see that this has kind of like a trigone appearance. It's going to be this little triangle. Here's that little triangle over here, that trigone. And that's going to be a triangle formed by the entrance of the 2 ureters. So the 2 ureters are actually going a little bit down. You see we have the ureters here. They're going down, 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 down. And they're entering into this area, right, into this hole over here, right? These are the openings of the ureters. So I'm going in through the back and then out through the urethra over here. And that gives us this whole triangle appearance. Pyramids are going to be in the kidney, excuse me. Okay. And if we're looking over here, we can kind of tell that this is going to be a male bladder. We can see that from the existence and presence of a prostate gland, right? So we have a prostate gland in males and not in females. We'll talk a lot more about the actual urethra and the structures when we get into the reproductive system. But for now, all you should really know is that there are a few differences between the male and female urethra, which we'll cover in just a moment. Okay, so here's a general idea of where the urinary bladder is located in male and female specimens of the human. So here we have the urethra, right? If we're looking at the urethra, we can see it's a lot longer in males, of course, due to the existence of the penis, which is going to be making it a much longer tube than in the female urethra, which is actually quite short, right? You have all of this length of the urethra over here going from the urinary bladder and then finally out to the environment. And then on the female side, you have only this small little stretch of urethra exiting into the environment. And that actually is one of the reasons that UTIs, urinary tract infections, are a lot more prevalent, a lot more common in the female population than in males. It's because there is a lot less distance for bacteria from the outside world to travel, right? So, granted it happens in both, of course, but it's just a little bit easier for a woman to get it than a male for that reason, because of that structural difference. Nothing to do with lifestyle or anything of that nature. Okay, then here, of course, is that urinary bladder over here on both sides. We can see that the uterus over here on the female is going to be directly superior to the urinary bladder, which kind of lends credence and legitimacy to the fact that women who are pregnant, right, when you have a child developing or a fetus developing within the uterus, you're going to see that uterus obviously expand to greater and greater volume sizes as the fetus develops. And due to that, you're going to have a little bit of compression over here onto the urinary bladder, which kind of is the reason pregnant women always have to pee, right? And it's like, it's not their fault. It's like literally their bladder is being pressed from the inside. So that kind of makes a little bit of sense. We'll talk a little bit more about all this in the reproductive section, but otherwise we can see that most of the other structures here, right? You have the rectum, you have the colon here, right? Same thing, same idea in the male and female anatomy, right? You can see that the males have the testes here, and the women are going to have the ovaries instead. All right, we'll talk about those. And here are ureters. Ureters are the same deal, all right? So just be aware of these minor differences as we go forward. Talking about the urethra briefly, we are going to have the urethra bordered by the two urethral sphincters. Okay, you're going to have an internal urethral sphincter, which is going to be smooth muscle, and that is one that we don't have voluntary control of, right? So here's in the female, we're going to have the internal urethral sphincter right up here at the junction between the bladder and the urethra. And in males, we'll have the same thing. We'll have an internal urethral sphincter at the junction between the bladder and the urethra. And the external urethral sphincter is going to be the one that is skeletal muscle. What does that mean to us? That means it is somatically controlled. If we have voluntary control over it, that's why you can kind of hold your pee if you really have to go for a long period of time. Right? Eventually, it'll give out, and you'll just, that's that. And that's why in a lot of, for example, geriatric cases, or you might have, muscular weakness conditions where you might notice incontinence in the patient. And that is, it's no fault of their own. It's because the muscles are weak in those sphincters, and that, no matter how hard they try or how much they want to, they simply lack control over those sphincters. And that ultimately leads to sometimes urinary incontinence, which, I would hope any of us in the healthcare field or working with patients are sensitive to, because, these folks don't, it can be a bit unsettling, a little bit embarrassing. And it's important, that we remain sensitive to these issues that are frankly out of their control, right? Anyway, I digress. That's beyond the point. External urethral sphincter, we can see that it is going to be over here in females, right? And notice the close proximity of the internal and the external urethral sphincter, right? And then here is the external urethral sphincter for males. Similar proximity, right? But the actual urethra is going to extend longer due to the its necessity to travel through the male penis. Okay, and we'll talk a little bit more about the surrounding structures as well. So that, you know, in the corpus cavernosum, corpus spongiosa of the penis, and when we get into the reproductive section. Okay. One thing to also be aware of in the male urethra is that it has 3 portions rather than just the female urethra, which only has 1 portion over here, right? The male urethra is going to have the prostatic urethra, okay? That's going to be this area over here. So this whole green line is going to be your prostatic urethra. We're going to have what's known as the membranous urethra, okay? That's going to be kind of, let me get my color here, right around here. And then finally, you're going to have the penile urethra, which is going to extend through the length of the penis, okay? And here I kind of just included just some images of some of the models that we have in the lab. You can expect these to be on your practical, right? You will definitely have these models on your practical, you know, these and similar models that you should be very familiar with. So as we all know by now, I'm not going to stress it too much because we are all definitely aware by now that in lab time is key, right? Practice with these models, you know, getting your hands on different perspectives and different models, and you know, using the slides that are provided in class, textbook, you know, for different information, different images, different diagrams. That is key, right? Of course, you want to get some time with the microscopes, looking at different slides, because we know that not all slides are going to be these picture perfect images that we're seeing on the PowerPoints, right, that are being distributed. So, you know, that is the answer. If you want to know how do I best prepare for this exam, get into the lab, okay? It's usually pretty empty, except for when we have classes going on, but there's only like 3 or 4 going on in that room. So, you know, definitely get some time in the lab if you can. Okay, now let's jump into histology. We'll talk about some major histological structures and landmarks that you should be aware of in these different structures that we talked about. Starting with, of course, the kidney, right? So here we are looking at the kidney. We can see that there is the cortex and the medulla. We have that cortex on the outside. And, you know, if you look at it in the top right-hand corner, you can kind of see how it's a bit lighter colored than the medulla inside, right? And if we look a little bit closer, we can kind of make out these little renal pyramids, right, that we saw kind of in the diagrams that we were talking about. We have the minor calyx over here, right, and that's going to lead into, of course, our major calyces, and then ultimately the renal pelvis and out into the ureter. Okay, here's another image, right? So we can see that the cortex is going to be on the outer portion, medulla on the inner portion, and that, you know, that black line is going to be the approximate border between the two. We know that there is a fibrous connective tissue capsule surrounding the entire cortex, the entire kidney, and you can't really see that at this magnification, but it is there. If we zoom in a lot, we'll be able to see it, right? So here we go. If we zoom in, now we can kind of get a better look, right? If you look closely, you can see a little bit of that border is going to be the capsule that we talked about. Okay, again, here's the medulla and here's the cortex. Now, let me point something out to you guys really quickly. That remember when we said the glomerulus and the Bowman's capsules are only going to be present in the cortex? Now, that's something that you should be looking at and looking for when trying to orient yourselves with a slide, right? So if we look at this and we know that it's a kidney and we want to know which parts are which, right, we are going to be looking for these glomeruli, for these renal corpuscles, right, which is the glomerulus plus the Bowman's capsule. So here's one, here's one, here's one, here's one, right, here's one. And these are going to be indicative of whether we're in the cortex or in the medulla. The medulla, we see a lot more of this, these long striations kind of going downwards toward the renal pelvis, which makes sense, right? That's going to have a lot of the longer ascending and descending loops of Henle, the collecting ducts, right? So that makes a lot of sense. And here we go. We zoomed in even a little bit more, right? And we can definitely see a lot more clearly these renal corpuscles, right? We're at 100x right now. And if you look closely, all these black dots are pointing out your renal corpuscles, okay? And you can kind of see the Bowman's capsule surrounding them in white over here, okay? Another view, okay? We have the glomerulus, which is going to be all in here. This is all the glomerulus. Right? The Bowman's capsule is indicated by the blue arrows. The Bowman's space, that little white space, is going to be where we're going to have the urine kind of coming out, right? So we're going to have the urine coming out of the glomerulus and entering into the Bowman's space, which eventually will enter into the proximal convoluted tubule, right? And if we look at what is staining really dark purple over here, right, some of these really dark purple cells that we're looking at, those are going to be podocytes, okay? We see them both in the Bowman's capsule and surrounding the actual glomerular capillaries. Here's another view of those, right? Bowman's capsule, that white part is the Bowman's space, okay, glomerulus. We can kind of make out some of the renal tubules over here in the bottom left, right? So the tubular region of the nephron is, we know it's composed of the proximal convoluted tubule, the thick and thin segments of the nephron loop, right, the loop of Henle, and the distal convoluted tubule. The proximal, the PCT, the proximal convoluted tubule, is the longest and most convoluted segment. So the majority of the tubules found in the cortex are going to be PCT segments, right? And that's going to be composed of our simple cuboidal or short columnar, sometimes, epithelial cells. But I'm just telling you guys this so you're familiar with what you're looking at. I'm not going to test you on specific tubules. But, it's important that you're familiar with the general makeup of a slide, right? Now, the distal convoluted tubule is going to be also simple cuboidal epithelium, but they're going to stain a little bit less intensely, a little bit less intense than the proximal convoluted tubule, right? A little bit lighter colored. The PCT, the proximal, will stain more intensely because it's pretty acidophilic, okay? Just so you guys are aware. And actually, here's a nice little representation of what I'm talking about. At the bottom here, you can kind of see a numerical indication of what we're looking at. So there's definitely a distinction between the collecting ducts and the thick and thin descending loops of Henle, right? And the capillaries. So you can look at those for your own interest. But for example, the collecting ducts, number 3 over here, they have very clear lateral borders distinguishing, right? These are the lateral borders separating cell from cell, right? These are a lot more distinct in 3 than they are in, for example, 2, or in, of course, obviously in 1, but those are your thin loops of Henle, right? Okay. So just so you guys know what you're looking at. Okay, so one more time, let's just really quickly run through the excretory passages, all right? So we know that There's going to be a series of passages that carry, store, and expel urine, starting with the minor calyces in the kidney, right? Those are going to be, multiple will be giving or merging to form a major calyx, right? The major calyces will then converge at the renal pelvis, and then the renal pelvis is going to transition into the ureter at the hilum, which will then empty into the bladder and exit through the urethra, okay? One thing I want everyone to be aware of when looking at these different excretory passages, these different urinary structures, except for the kidney, is that they're going to be lined with transitional epithelium, and sometimes it's referred to as urothelium. And this is a very specialized type of stratified squamous epithelium that is impermeable to electrolytes and water, which, makes sense. The nephron is going to be permeable to extra electrolytes and water, different parts of the tubules, but we don't, by the time it enters into the ureters, we're done. We're done with all of the filtration processes and all of that. So we're just transporting and storing after we leave the kidney. All right, so these structures are all going to be impermeable to electrolytes and water due to this specialized urothelium. And we're going to see, as we saw before, that they're capable of flattening to accommodate increased fluid volume in the luminal space, especially in the bladder, right? The bladder is going to be very accommodating. It can expand to great volumes and shrink back down to a very small size, right? And then we're going to see that the most superficial layer will be referred to as dome cells, just because of that dome-like appearance that we saw earlier. Specifically, that's only going to be found in our non-distended lumens, right? Now the lumen is that opening, that little space in the middle, so, right, in a non-distended ureter, a non-distended bladder, a non-distended urethra, that's when we'd see those dome-like superficial epithelial cells. Okay, so we left the kidney. Let's talk a little bit about the ureter now, the ureter. So the ureter and the renal pelvis and all of its subdivisions are going to constitute the main excretory ducts of the kidney. All right, the walls of the ureter are composed of 3 layers. We're going to have the inner mucosa, a middle muscularis, and an outer adventitia. Because of the high content of elastic fibers within the wall of the ureter, the lumen is quite often going to take on this star-like appearance, right? A stellate shape, okay? And that is going to be pretty common in ureters especially. Now, the mucosa of the ureter is going to be lined by that transitional epithelium, right? We talked about that. And that epithelium, as we all know, epithelial layers are going to be sitting on a thin basal lamina, and you rarely can see it. You really can't see it on a light microscope. It's very tough to discern, but it's there. Now, the ureter, in terms of the middle muscularis external layer, is going to be different in certain sections. Now, the upper 2/3, right, the proximal 2/3 of the ureter, that's going to be composed of 2 layers of smooth muscle. You're going to have an inner longitudinal layer and an outer circular layer, right? So the longitudinal layer is going to be going more up and down along the length of the urethra, and the outer circular layer is going to be going kind of as a ring around the tube of the urethra, of the ureter, excuse me. Okay, that's for the upper 2/3. Now the inferior, right, the distal 1/3 of the ureter, you might see an additional layer present, and that's going to be a kind of a discontinuous outer longitudinal layer of smooth muscle, right? So you would see in that bottom third an inner longitudinal, an outer circular, or a middle circular, and then an outer longitudinal layer. So it'll be a middle circular layer kind of sandwiched in between 2 longitudinal layers, okay? And then finally, that adventitia is going to be composed of areolar connective tissue with a lot of blood vessels and structures like that. So, it might not be present in all sections, but, on the one on the left, for example, we can see that very clearly. We have a nice adventitia, very loose areolar connective tissue, right? And some adipocytes surrounding it. And that's usually pretty distinguishable from the muscularis layer. Okay. So let's take a look over here at the ureter again from a different perspective. We have that star-shaped lumen and the lining transitional epithelium. These are very distinctive features of the ureter. So definitely take a moment to examine the epithelium a little bit. The smooth muscle in the muscularis layer is arranged in these distinct circular and longitudinal layers, right? So this obviously is going to be a section from the bottom third of the ureter, because we have three distinct muscle layers. We have the inner longitudinal smooth muscle layer, that middle circular smooth muscle layer, and the outer longitudinal smooth muscle layer. And how can we tell the difference between these different layers? It's the orientation of the muscle fibers, right? So we know by now that the longitudinal sections, right, are going to have more like dot-like appearances, kind of like dots, right? You can see the nuclei, kind of the actual individual muscle fibers in the inner and outer longitudinal muscle fibers, muscle layers. And then in the middle is going to be more of kind of like a circular type of vibe. It's going to look circular, right? Because it's just based on the sectioning that is going to make that distinction, okay? So again, the ureters will have those three coats: the mucous membrane, mucosal membrane consisting of the transitional epithelium, usually about 4 to 5 cells thick, in longitudinal folds that are going to be supported by a lamina propria of dense connective tissue. And we can see that if we zoom in a little bit over here, right, we can see these transitional epithelia. They're kind of dome-shaped up here. And then they're going to have the lamina propria directly underlying it, right, that dense connective tissue. And then finally, we can see that muscle coating, that muscularis externa, with our inner longitudinal and outer circular muscle fibers, right? And that's going to be in the top 2/3 of the ureter. And then if we were in the bottom third, we would see another outermost layer of longitudinal muscle fibers. Okay, and then finally we have the adventitia, which you can kind of see in this zoomed out version over here, just that loose connective tissue. So here's one particular slide of the ureter, right? This is what you might see under a microscope. Here is that inner lumen that we talked about. So if I were to zoom in on the luminal aspect, right, on some of this transitional epithelium pointing toward the lumen, I would be able to see this transitional epithelium, right? And given the relatively dome-shaped appearance that I'm seeing over here, it's not too flat, right? I can probably surmise that we're looking at a relatively empty ureter. It's not too full, right? This is going to be a lot more apparent in the bladder than it would be in the ureters, but it's still noticeable, right? If we have this stellate appearance in the lumen, it usually tends to mean that we're looking at something that's not totally full. If we zoom in on this area over here, so here, we have this, and we're going to zoom in now onto the muscularis externa layer, and that's going to show us kind of an irregular arrangement of smooth muscle in two layers, right? You have the inner longitudinal and the outer circular layer, and that's going to be in the upper ureter, and then the three different layers in the bottom. Now these are very irregular arrangements, guys, so don't go crazy trying to discern, you know, specifically which layers of the muscularis are present. Just be able to know whether there are two or three. Okay. And then finally we have the adventitia. And that adventitia is going to be loose connective tissue with some blood vessels. Right, we're going to see all kinds of different, different structures of that nature. That's going to be over here on this next slide, excuse me. All down here, kind of spread out. Okay, we can see another image on this next H&E stain, this next slide, which is going to show our nice longitudinal smooth muscle layers, and then our circular smooth muscle layers. We can see that transitional epithelium, right, supported by the lamina propria. Okay. Okay, now let's take a look at the bladder. This is actually the bladder. Notice it's very similar in appearance to the ureters, and the ureter does empty urine into the bladder, and that transitional epithelium is going to continue over the surface of this organ. Okay, so the thickened muscular layers are going to become interwoven, and they can't really be clearly identified at this point, but they're there, right? And it's going to be a lot thicker of a muscularis layer in this bladder. Okay, this image is showing a relaxed bladder where the epithelial cells appear a lot more dome-shaped, right? You can see a lot clearer that these are domed, and if they were, if it was distended and the bladder was full, we would see that these dome-shaped epithelial cells would be a lot more flattened and squamous in appearance, right? So that lumen, like we said, is going to be lined with transitional epithelium in the bladder. It's very well adapted to the lining of the urinary tract. It can tolerate repeated cycles of stretching and recoiling, right? So filling, right, and emptying. And that muscularis layer is going to be a lot thicker, like we said, in the bladder. So let's take a look. Here we have that transitional epithelium over here. The mucosal folding is pretty variable depending on the state of the organ, whether it's distended or not, right? That depends on when the slide was made, right? This is called fixation, so whenever the slide was fixated. The epithelium is not going to be uniform in thickness, right? And that is kind of owing to that variable distension of the organ. And although it might be difficult to distinguish, the muscularis is always going to be composed of 3 layers. And, it is hard to tell which one is which layer is which. But we can always see the difference between the bladder and the ureter because of how thick that muscularis layer is. That's one of the main distinguishing features between the bladder and the other organs, right? So here's a little slide of the bladder you might see under a microscope, okay? We can kind of see the top luminal surface up there, right? So if we zoom in on that luminal surface, we're going to see the transitional epithelium. We can see those dome-shaped appearances of the top superficial layer, right? If we zoom in over a little bit closer under here, we can see the lamina propria, that thick layer of dense irregular connective tissue that is underlying the epithelial layer. It's going to be a connective tissue rich in collagen and elastic fibers and whatnot. So, if you're not familiar already with, being able to distinguish between muscle tissue and connective tissue and epithelial tissue, I would definitely go back and take a look at your old lecture notes from AMP1, because that will come in handy when it comes to this histological identification of the urinary system, or of any system for that matter. Okay, so if we zoom in now a little bit, here's that little blue box that we're zooming in on from the whole slide. If we zoom in now, we can see the muscularis layer, right? And that's going to be this very big. Notice the size of the blue box relative to the entire slide is huge, right? So we have the muscularis layer, and it's hard to discern exactly which layers are which, but there are 3, and it's just super thick, okay? And that's the key thing that I need you to take away from this, okay? If we were, however, we could discern them. For example, here, let me zoom in a little bit on this one portion. Look at the blue box on the bottom right for reference. This is going to be that inner longitudinal layer, and you can see that from the orientation of the fibers, right? They're kind of going. There's a lot of little dots kind of pointing downward, right? That inner longitudinal layer. Now, if I were to zoom in on this middle circular layer, right? So that's blue box over here. This middle circular layer has a lot more of the that kind of flowy appearance to it, right? It's going to be having a lot fewer dots, a lot fewer nuclei immediately present and visible. You can't really see the individual muscle fibers, but you can see the directional orientation of the fibers going from side to side more than up and down, right? And then if we go down here in this blue box, we can kind of come back and see that inner longitudinal layer, right? Or that outer longitudinal layer, excuse me, which is going to be a lot more similar to what we were talking about before, right? We're going to have these individual muscle fiber bundles. You can see the individual fascicles kind of going up and down over here, right? And then finally, if we go all the way down to the opposite side of the lumen, we can see this adventitia, which is going to just be that loose connective areolar tissue. It's going to have some blood vessels and some underlying smaller structures in there. A lot more light colored, right? Okay, here's that transitional epithelium again, right? We already talked about the difference between the tissue and how it looks when it's distended versus empty. And now we can jump right into the urethra. Okay, so the urethra is going to have, again, 3 layers. It's going to be the mucosa, right, which is going to be the epithelial tissue. It'll have the muscularis and the adventitia. Now, I'm not going to go too much into the urethra on this lecture. We're going to spend some more time on the histological differences between male and female when we get into the reproductive unit. But for now, just be familiar with the fact that it kind of has a similar looking appearance, not as much of a muscularis layer as we saw in the bladder. And we're going to see also a little bit of a change in the epithelial mucosal layer, right? So we're going to start off with transitional at the more proximal portion of the urethra. Then it's eventually going to transition into, no pun intended, it's going to eventually turn into pseudostratified columnar epithelium. And then from there, it'll become stratified squamous epithelium. Okay, I'm not going to get too much into that, but that is kind of an FYI for you to be aware of. And that pretty much covers it for the histological portion of today's discussion. Okay, I put in a few images from the Mink identification PDF that I have online. So if we look into here, we can see that, you know, obviously the digestive tract has been removed for clarity. Remember that this is a retroperitoneal organ, the kidneys, right? So they're going to be behind the peritoneum, which is going to be holding all of these different organs that would normally be in the way. But we can see the right and left kidney over here pretty clearly. And from there, we can take a look at the entrance of the renal arteries and veins, right? And the exit, or the entrance of the renal artery and the exit of the renal veins and of the ureters over here. Now, the best way that you guys can be able to identify, because there's a lot of little tube-like cords kind of all over the place here, right? You got suspensory ligaments, you got different things, you know, vessels, you got nerves, all kinds of stuff that, you know, we can't be 100% sure with the naked eye. So the best way for you to be able to identify is by backtracking, right? Backtrack on your step. What is coming out of where? What is entering into what, right? So if we look at this renal hilum, that hilum where we have all these important structures entering and exiting, right? The renal artery and vein are relatively straightforward. Fortunately, we have, you know, latex, colored latex that we use on the minks, so we can pretty easily distinguish between the inferior vena cava and the descending aorta, right? The descending aorta is going to be a lot thinner than the vena cava in most cases, and we can see where the renal vein is dumping into the vena cava, and we can see where the descending aorta is branching into the renal artery. The ureters, however, we can see are also coming right out of that hilum, right? They're coming right out of the hilum of the kidneys, and they are clearly leading into this one organ, which must be the urinary bladder. So it might seem a little bit, tougher on an actual living specimen to be able to identify these things, but it really just comes down to being able to connect the dots in terms of what is entering into where, what is leaving into where, and that just takes, a pretty straightforward understanding of the anatomical structures of the urinary system, right? So here's another look of the kidneys that have been isolated, right? They've been removed from the specimen itself, and we can get a much better look at the renal vein and the renal artery and the ureters leaving the kidney, right? The veins leaving the kidney and the renal arteries entering into the kidney, okay? So this should be a pretty good look at what we're talking about, okay? And here we go again with a sectioned view of the kidney itself. We can see the renal pelvis over here, which is where everything is going to be kind of emptying out into, right? We have the different calyces you can't really see the calyces too well on this, but you have the minor calyx and many of those emptying into the major calyx. Many major calyces are going to empty into this renal pelvis, which we can see nicely. And then that renal pelvis is going to dump into the ureter. Okay. We can see how some of the different vessels are distributed all throughout. We have that renal artery here, okay, coming on in. It's kind of hard to see any of the actual renal or the arcuate arteries themselves, but you can see the general arcuate vessels kind of up in this general area over here. And so these are probably going to be more identification on models, right, for these very, very small vessels, but they are present. So you should be able to kind of see that there are interlobular, interlobar, and, arcuate vessels, both veins, and, you can see the segmental artery a little bit over here on the bottom, right? There's no segmental vein, remember that. So just get a general idea. Go online as well and look up different MINC images if you like. You can look up different histological slides. There are tons of websites, excellent websites, where you can really zoom in like on your computer, just like you would on a microscope, and just get a much clearer understanding of different slides and what they look like. Okay. And we can call it there. I think that'll cover it for now for our urinary system. And I will see you next week.